Thanks, Alex, and good morning also from my side. And thanks for being here at the fourth day, this bit of the intense program. So in the past days, you have heard a lot about surface EMG, and you also have heard that surface EMG is only able to detect the activity of superficial motor unit. So if we want to go beyond the surface, then we need to use different type of sensors that we place within the muscle, and this is why we talk about intramuscular EMG. So my research focuses on understanding and developing technology to be able to record the activity of a large population of motor neurons. And the reason why we believe that this is important is because in this way, we believe we are able to link cellular behavior to functional behavior. And as I say, in order to do that, we have developed new technology. But to understand why this is needed, I will talk a little bit about conventional technology that is used to record intramuscular EMG, what we have developed, and finally, I will briefly discuss about some applications, both in fundamental research, try to understand the principle of motor control, but also in the field of neural interfacing. Now, you are, the first slide has a bit of summary and very basic knowledge that at the fourth day of the summer school you already have, so I will be quite fast. So you know that when we decide to move, signals travel from the brain to the spinal cord. At the spinal cord, we have, this doesn't work again. We have pools of motor neurons. They exit the spinal cord with their axons and they synapse into muscles. So the next slide, you have a bigger view of the motor neuron and the muscle they innervate. And on the right side, you actually have an animal slice where you can see what is represented in the cartoon. We have the nerve that reaches the motor fibers, the muscle fibers, and synapses into the fiber at the neuromuscular junction. You have also learned that motor neuron innervate a set of fibers, and we refer to motor unit as motor neuron plus the fibers that they innervate. Now, summarizing what I say so far, muscle are made up by motor units. These motor units are located at the spinal cord. And the problem is that it's very difficult to access those motor neurons because the spinal cord is well encased within the spine. So how can we record the activity from motor neurons? So a motor neuron is an integrator, so it receives input from supraspinal center, afferent feedback, and the motor neuron integrates this feedback. When the current is above a certain threshold, it discharges an action potential that reaches the muscle fiber at the neuromuscular junction, and then the action potential travels towards the two tendons. So if we place an electrode, this electrode will record a motor unit action potential. So each motor neuron action potential becomes a motor unit action potential, and it's thanks to this one-to-one correspondence that we can record from the muscle, but retrieve the activity from the spinal cord if we have appropriate signal processing methods. What do we want to extract from these signals that we record from the muscle? Basically, the same variables that our central nervous system uses to regulate force. So here you have a person who is holding a ball and the weight increases. And what you can see is that more and more motor units are recruited from the small one to the big one. This is the Henman principle we have heard a lot about. So recruitment is the first variable used by our central nervous system. And the second one is discharge rate. The faster a motor unit discharges, the more the force that is exerted. So the recruitment and discharge rates, these are the two variables we are interested in. And how do you extract that? Again, this is a slide you have seen in some previous presentation. The EMG that we record with our sensor is nothing else than the sum of action potentials. It can look like that. It's very sparse in case of intramuscular signal. It's much more interference in case of surface EMG. And what we want to do, we want to reverse engineering the EMG generation process and retrieve actually this variable through decomposition. So these variables are those who contain the neural information, so how many units are active and when they discharge. So how do we do that? So in these days, you have learned a lot about surface EMG, and probably you are much more familiar with that. This technology is very in fashion now, but this is not how everything started. So for more than 100 years, for about 100 years, we have been recording the activity of motor units using invasive electrodes. These electrodes are very selective. So when you insert them into the muscle, you get a signal that is very sparse. And in this condition, you can go back to the activity of individual motor units. The first recording from muscle fibers was done in 1928 using a concentric needle. So again, this type of sensor is very selective, so you can actually see the activity of individual motor units. Then 30 years later, Van Bejan and Stecco proposed the use of fine wires. So fine wires are also very selective, but they are much more flexible and therefore, hopefully, less uncomfortable for the study participants. And in their paper, they also explain how you can build them. So you have here a metal a metal wire, and this metal is covered with some insulation, for instance, nylon in this case. So you make a loop, you insert into an hypodermic needle, then you remove the insulation at the tip so that the bare metal is actually able to record the activity from the muscle. You cut this loop so that you create two wires. You bend them so that you have two barbs. And these two pieces of metal are those that will be able to record the signal when you insert in the muscle. Now, different from the concentric needle, in this case, the needle is just an intruder. So use the needle to insert the wire into the muscle. Then you remove the needle, the wire stays within the muscle and they record the activity. Now, as I mentioned, these electrodes are very selective and they are purposely selective. In this way, when you insert them in the muscle, so this pink oval is the cross-section of a muscle and this circle are motor units. So since the electrodes are very selective, you pick up the activity of few units, 2, 3, 4. So in this condition, even by visual inspections, you are able to actually detect the activity of individual units. And so what you want to achieve with a recording is a signal that is very sparse. And this signal is very sparse, as I say, because the electrode is selective, but also for another reason. Because you place the electrode within the muscle. So in this cartoon, you have here the muscle, a layer of fat, and the skin. So what you have seen in the previous phase is that you place the electrode on the skin here, and what you get are potentials that are smooth, and also they have a large time support. But if you record directly from the muscle, your action potential will be much more spiky, and also the time support will be much narrow. So this is why you have less overlap, and this is why intramuscular is another reason why intramuscular EMG is so sparse. So basically, these are potentials recorded within the muscle. When you place the electrode on top of the skin, like the fat layer and the skin act as a low-pass filter, and they smooth the action potentials so that they become, they get a larger time support. Now, how do we actually do this sorting, this decomposition when the signals are so sparse? You have heard a lot in the past days about blind source separation, but you know that blind source separation needs many channels to be applied, while in this case, we have only one channel with conventional technology. So the signal processing pipeline is a bit different. You normally start pre-processing the signal, then you segment them, you do some clustering, and the last step is to resolve a superimposition. And I will show this processing pipeline using an example of a signal that I took from the EMG lab website. This signal was recorded from the brachioradialis muscle using 4 invasive electrodes, but I will just show one channel. And this is where the signal was taken from. When I showed the processing pipeline, I actually took a screenshot from the EMG lab software that was made available to the community to be able to decompose intramuscular signals. And it's a great software that we have used a lot in our studies. So first step, as I mentioned, is pre-processing. This is on top here. You have a raw signal that has been recorded with invasive electrodes. And you can see that usually it's flat, but from time to time you have some activity. The first step, pre-processing, consists in applying a high-pass filter. Why do we do that? When we apply the filter, you see that the action potentials becomes much more spiky and they stand out from the background. So it's much easier to process this type of signal. Then we need to segment. Segment means that we actually go through the signal and we detect all portion of the signal where there is activity on top of the background noise. So here, for instance, I detected the first 13 active segments. Now, if you look at this segment, what you need to do next is cluster them into different motor units. Now, if you look at this image again, you can see that there are some waveforms that they repeat consistently. For instance, in red, you have active segments with a very similar waveform. They belong to the same motor units. And so the red one is the first one, but you also have the green one, the orange one, and the blue one. And you also have some outliers, such as the waveform 4, that is similar to some of the units, but is not exactly the same. So for the moment, we ignore it. Now, if we process the whole signal, you will be able to recognize 4 clusters corresponding to the 4 different motor units. And I represented here in next slide. So this is a superimposition of action potentials belonging to the same motor units. This representation is usually referred to as shimmer plot. And if you take an average, you actually get the action potential template. Now, what you have in this panel is the discharge pattern. You have seen a lot of raster plots in the previous presentation. Here, we represent each discharge with 1 point. Now, this signal was recorded during an isometric contraction at a constant force. And you know from physiology that in this type of contraction, usually the action potential discharge regularly. But you can see that these firing patterns are not regular. And why is that? It's because we have ignored sorting the outliers. And this is the final step to achieve a complete decomposition. And this last step is usually referred to as a superposition resolution. So if we go back to our outlier, the black waveform, we can see that actually contains both the red waveform and the blue waveform with an appropriate shift. So resolving the superposition means that actually in this compound action potential, you need to understand which are the components. And once you do that for all outliers, what you get is a complete firing pattern. Now, this signal, is very sparse, and I hope I convince you that even by visual inspection, you can sort it, which is something you cannot do with surface EMG. But another way to be convinced that you have done your decomposition well is actually to take a low-pass filter. And what you notice is that you have common fluctuations in all the instantaneous discharge rates. This is due to common drive. You heard a lot since the day one. So due to the common drive that a pool of motor neurons receive, the result is that the discharge rate have this common fluctuation. So this is a way for you to understand, to be more confident about your decomposition. And of course, it's something that you can apply not only to intramuscular detected motor unit, but also to surface, to motor units detected from the surface. Now, I did this process manually, but of course there are softwares that help you in the decomposition process. For instance, EMG Lab is based on template matching. So the method is semi-automatic. You get some decomposition automatically, and then you need to edit. The process is a bit different from surface EMG, but the aim is the same. Which are pros and cons of these intramuscular electrodes? So a good thing is that you can actually detect the activity of motor units. You can perform the decomposition. And you can also do it via visual inspection if the signal is sparse enough. So if the electrode is selective and if the force level is not too high. For this reason, intramuscular EMG is considered the gold standard and has also been used to actually validate the decomposition of surface signals. But there are also some disadvantages. Being the electrode very selective, you can detect the activity of few units only. And because of this limitation, actually, this limitation somehow motivate the development of new technology, which I will discuss in the next session. So why we are not very happy with the detection of few motor units? So in this table, you can see the number of motor units or motor neurons that innervate a muscle. And you can see that from some muscles, like first total interosseous, is about 100, but for really big muscle, like leg muscle, like this one, for instance, you have 4,000 units, right? So this is a huge number compared to 3, 4, 5 that we can detect with intramuscular EMG. So our question is, I mean, when we detect these 3, 4, are we actually getting information that is representative of the whole muscle? Maybe yes, maybe not. So people have tried to address this issue in different ways. And this is an elegant study from the Neuroscience Research Australia Institute. These people use, like researchers use intramuscular EMG to detect activity from the genioglottis muscle. So I don't know if you are familiar with this muscle. It's a muscle that controls the tongue, and it's a muscle that we use to stick our tongue out. But this muscle is very important also when we sleep because it maintains the airways open. So if the muscle doesn't work properly, we actually experience apneas. So this is one of the reasons why it's interesting to study this muscle. You see here that the muscle is deep. So there is another muscle before. So it's not possible to access it with surface EMG. We really need to use a needle. And what people have done in order to increase the number of units that were detected was to insert the needle 3 times in each study participant. And once it's within, like they manipulated, they tilted to plus minus 30 degrees in order to change the position and detect the activity of more units. This may be a bit uncomfortable for the subject, of course, but also from a signal processing point of view, you cannot do any population analysis because these are serial recordings. So the activity of the motor units that you are recording are active one after the others, not at the same time. Then this is a good solution for some type of analysis, but maybe not appropriate for other analysis. Then this is why people have thought about somehow advancing the technology. And one of the first advances was the use of high-density surface EMG for motor unit detections. So Alex was pioneer on that, and he developed his famous CKC algorithm to actually be able to decompose high-density surface EMG signals into the motor unit action potentials. Now, I will be very brief on that because you have heard a lot in the past days. Also, this technology has pros and cons. So the advantage is that it's not invasive. So you can use it also with children, with patients, with people who are afraid of needles. But another advantage is that you can detect many more motor units compared to intramuscular EMG. But there are also some disadvantages. So we can use the surface EMG also to be able to record from superficial muscles. And you have heard many times in the past days that the units that you detect are the units that are more superficial and they have a large action potential. So what you have in this image are some simulated signal, the dark one. And this signal have been obtained by these motor units that are active. So all the circles represent motor units that are active. Now the circles that are filled represent the motor units that the surface decomposition algorithm is able to decompose. So what you can see is that these units are close to the surface and they are relatively big. So basically only those units who have large surface charge potentials are detected by surface EMG. Okay, so what we get is a biased sample of the motor unit constituting the muscle. This is why we thought we need to further or to develop even other types of technology in order to be able to solve these problems. This is the same slide that you have seen for conventional intramuscular EMG. And what you have seen here is that these signals are very sparse. What we thought was, why not to build an electrode that contains small detection sites, similar to normal intramuscular EMG, so that the signal is still very selective. But if we place many electrodes within the same wire, we are actually able to perform a spatial sampling of the muscle electrical activity. And I apologize. This is the first prototype that we developed. It's a wire, very thin wire, just 20 micron thick. And we place 16 detection sites in this electrode. These are made in gold and platinum. They are very small as a normal intramuscular electrode. And you can actually see here, actually they are so small that probably you cannot even see them. And these 16 electrodes are in this wire and are inserted into the muscle with a cannula as a normal intramuscular EMG, a conventional one. So once the electrode is inserted, we remove the cannula and the wire stays inside for recordings. Now, if we design this electrode properly, we can actually record signals from the whole cross-section of the muscle, so we can detect the activity of more units. And this is one of the first examples, signals detected from the abductor digital minimum, so the muscle that we use to perform this movement. And you can see that we can detect different units from different channels. So as a result, we can increase the number of motor units that we detect. Which are pros and cons for this technology? So we can detect many units. We can detect both small and big units, but also in this case, like we have some negative sides. So these electrodes are invasive, even if minimally. And if you process them with plate matching techniques, you actually need to spend much more time. I don't know if any of you have ever done spike sorting, but it's a very time-consuming job. And of course, if you increase the number of channels, you also increase the time you dedicate to process these signals. But you have heard a lot in the past days about blind source separation, and the mixing problem is similar. So the idea was, why not to apply blind source separation to our signals as well? Now, these algorithms you are very familiar with are actually defined blind because they don't make assumptions of the signals. You know nothing about the experimental protocol. You don't know how many units are in the signal. So this is a very good thing, but this algorithm have a very strong assumption that you need to have more EMG channels than motor neurons that are active. If this is not the case, what the algorithm does is just to pick up the activity of the action potentials with more energy. So when we have applied the blind source separation to our intramuscular signal, what did we get? So this slide shows different recordings. And this is the number of motor units that we have detected manually. So via template matching using EMG lab. And this is the number of units that blind source separation detects. Now, if we focus on the last row, you have 60 units against 21 units. So only 1/3 of the units were detected automatically. And this is because the number of channels is actually not too big. So next step was, why don't we increase the density of the channels? And this is what we have done in our latest development. So we have now an electrode where we have 40 detection sites distributed over 2 centimeters. And this is how the electrode looks like. Actually, here you are able to see the electrodes. It's very similar to the previous one that I showed you. The connector, of course, is much bigger because you have more channels that you need to record from. Now, these are signals or the result of decomposition applied to signals detected with these electrodes. So we had 2 electrodes inserted into the tibialis anterior muscle, so 80 channels, and you see here that we have more or less 80 motor units. So now we have a big sample, unbiased sample, so we get activity from both the surface and from both the large and small motor units. But something very important, we get much better results when we use automatic decomposition. So again, this slide is similar to the one you have seen before. Here we have units detected manually. I process the signals, and Alesh, you didn't know the results, my results decompose the signal automatically with the CKC algorithm. And you actually can see now that in this case, we are able to detect 75% of the units, not 30% as in the past. But also you can see that the accuracy of the automatic decomposition is very high. It's 99% in average. So basically, this signal didn't undergo any editing. So these are the result of the blind source separation algorithm. So the agreement was 99%, even without any editing. There were a few doublets that the algorithm could not detect, and this is why actually we didn't reach 100%. So summarizing, we have got these electrodes that contain an high density of detection sites. We can get EMG of good quality. We can decode the activity of large population of motor units, both superficial and deep. And what is important is also that we can tailor the design depending on the application. So our first electrode had 16 detection sites. Then we had one that could be used both for detecting EMG signals and stimulating. This is the electrode you heard from Alejandro yesterday. And our latest development includes an density of detection sites very close to each other. So which of the 2 methods is better? Should we use surface EMG or intramuscular EMG? So both methods have pros and cons. As we have seen, surface EMG can be only used in superficial muscles, but it can be used in all subjects, including very young people and people who are afraid or don't like to have the needles inserted into their muscle. Intramuscular EMG can be used to in both deep and superficial muscle. It's invasive, but it's a minimal invasive procedure. You place the needle, you remove it, and the wire is so thin that once it's within the muscle, you cannot perceive it. But of course, for instance, it cannot use in case of coagulation diseases. So what you should use really depends on the application. But since in the past days you have heard a lot about surface EMG, I will now show few applications, few experiments where we have applied electrodes in animal and humans. So these are humans experiments. And we have applied electrode in superficial muscles, such as the abductor digiti minimi, the tibialis anterior, but also in deep muscles, such as the genioglossus. And then we have used electrodes also in animal models, such as non-human primates and rats. So I will show few of these applications. So welcome back. In this second part, which will take half an hour more so that we have a lot of time for the quizzes, I will present a few applications. I think 2 or 3 in the field of motor unit physiology and 2 in the field of neural interfacing. So the first study that I want to show has been done at the Neuroscience Research Australia Institute, and is a similar protocol to the one I showed at the beginning. So in this case, we recorded from the genioglossus muscle while people were lying down and breathing quietly. And we have inserted 2 electrodes, one on the anterior part of the muscle and one in the posterior part of the muscle. And this is the results that we have obtained. So I'll try to explain this graph. So this line represents a respiratory cycle, and we have inspiration first, before the dashed line, and then expiration. So we breathe in and out. And each line represents a different motor units. Different colors represent different discharge rates. And when you have a continuous line, that means that this motor unit was active tonically for both the inspiration and expiration. And when you have a bold line, that means that here, this motor unit increased the discharge rate up to the maximum that was here, then decrease, and then back to tonic activity. So what you can see, like if we move here, we have the same image, but just the electrode are split. Those have been recorded from the anterior portion of the muscle, and you can see that these are units that are more active during the expiratory phase. While from the posterior part of the muscle, we have more units that are active during the inspiratory phase. Now, these results are somehow different from what was found in the literature up to this point. So these are data pulled from different subjects, and you actually see that most units were detected during the inspiratory times. And we believe that this is due to the way signals were recorded. You remember I told you at the beginning, what people do is like to insert a needle in the middle of the muscle, then they move it. But what happens is that you don't get the full story. Like if you want to get a comprehensive information about the whole muscle, it actually may be important to record from the whole muscle. So now this muscle is also very particular because you have inspiratory unit, expiratory unit, phasic unit, tonic unit. But I hope this example convinces you that like to perform a spatial sampling of the muscle, at least in some cases, can actually be very useful to understand what's happening. And another application where I think our electrode could be useful is in the detection of the territories of motor units. So this is done with a technique, conventionally, with a technique proposed by Stolberry and Anthony in the 80s. And how do they do that? So for each motor unit, they insert an invasive electrode that is so selective that can detect the activity from one motor unit only. And then they insert in the muscle and they slide into the muscle another electrode that is a bit less selective. And whenever you see an action potential, you know that you are within the territory of this motor unit. Then when you are done with a motor unit, you move this electrode, you insert this electrode again, you slide into the muscle, and you actually get the territory of another unit. Now, differently from this method, if once you insert the electrode and we perform a contraction, we can actually detect the territories. You see like this region where the action potential stand out from the background for all the motor units that you have detected. So you need just a needle insertion to calculate the territories of more units. So that again, from a subject point of view, may be less uncomfortable. Next recording, a recording that we have done with Robin Mildren and John Hopkins in Cullen Lab. This is a recording from postural neck muscle in non-human primates. And this is just a representative example. You can see that the area where each motor unit is active is very small. So you have these spatial localizations, and you can use, these are data obtained via decomposition and spike trigger averaging. And this type of representation helps you to understand if different portions of the muscle are active during different tasks. Now, next two applications are more related to myoelectric control. I apologize, it's not spelled properly. So control of neuroprosthesis. So this is a study hopefully almost accepted by Agnese Grisona at Imperial College. And what she has done is to use the electrodes, the high-density electrode that I just described, to record from forearm muscles. So you can see here the target, and this is an ultrasound image where you can actually see the tip of needle while entering the muscle. So as I mentioned before, this is just during the insertion. Then once the wire is within the muscle and you remove the needle, you won't be able to see the wire via ultrasound because it's very, very thin. It's like 20 microns. So this slide is just to show that, muscle of the forearm that are very small are another example where surface EMG may be less appropriate because they are so small that you'll have only few electrodes on top of each muscle. So in this case, intramuscular EMG could be a better choice. Then another application where we have used our electrodes is to somehow refine surgery models to improve myoelectric control. I don't know many of you works on the field and if you are familiar with the control of prosthesis, but what I have on this slide is the representation of a surgery technique proposed by Todd Kranken at the Rehabilitation Institute of Chicago and called targeted muscle reinnervation. So you have here a subject with an amputation that is very proximal at the shoulder level. So there is no arm left, and when you have a prosthesis, in this case, you need to be able to open and close the end of the prosthesis, to rotate, and also to flex and extend the elbow. So what you should be able to perform with the prosthesis are three functions. But you don't have many muscles left. So there is the pectoralis available that you normally use to control the shoulder, but the shoulder is not longer available, right? So you won't be using this muscle. So the idea was this brilliant, really genial idea from Chicago. Like our upper limb is controlled by three nerves. So we could split the pectoralis muscle in three portions, reinnervate each of these portions with one of the nerves that innervates the upper limb. And what happens is that this nerve reinnervates the muscle acts as a biological amplifier, and now you have three independent sites of control that you can use to control different functions of the prosthesis. For instance, with this, you control opening and closing. With this, you control rotation. And with this one, you control flexion and extension of the elbow. So this bit of having no muscles left, you are actually able to control a prosthesis with multiple degrees of freedom. So what we were interested in is try to understand what happens to the muscle when you actually do this from a physiological point of view. And I will show now a study where we just contributed with the electrophysiological recording, but surgery was done by the group of Professor Asman at the Medical University of Vienna. And what our colleagues, surgeons, have done is to reproduce the targeted muscle reinnervation model in a rat, so at the biceps. And what we have done is to place electrodes on top of the muscle. So now, so far, we have discussed about surface electrode and intramuscular electrodes. This recording were neither surface or intramuscular. What our colleagues have done is to open the skin and place the electrode directly on top of the muscle. So we refer to this recording as epimesial. So not on top of the skin, not within the muscle, but directly on top of the muscle. And the electrodes were aligned with the muscle fibers. So what we could do in this case is actually to record the signal propagation, the propagation of each motor unit. So if you focus on this image, you see that here you have an action potential that goes up first and then becomes negative. And this is the same for all action potentials that are on this side. While here, you have action potentials that are negative first and then positive. And this is true for the action potentials on this side. So this is because the change in polarity is because here you have the innervation zone, you have the neuromuscular junction. This is where the nerve enters the muscle, and then the signal travels toward one tendon and towards the other tendon. Okay, so like the change in polarity was help you to understand when the nerve enters the muscle. And you can also notice that the signal shifts to the right when we go far away from the nerve. This is because the signal takes some time to propagate along the muscle. So since we know that the distance between two electrodes is 1 millimeter, we can calculate the time it takes for the signal to propagate from this electrode to this electrode, and we can actually calculate the conduction velocity, so how fast the signal propagates. And a conduction velocity is also a measure of motor unit size. So we could actually check how the composition of the muscle changes after reinnervation. Rather than the physiological data, I will actually show the imaging data from our colleague Konstantin Bergmeister. So what you have here is a bicep muscle. And you can see that this muscle is composed by type 1, type 2A, and type 2 fibers, represented in different colors. While if you take this biceps, and this is an intrinsic muscle of the hand. In this case, you have type 1 and type 2A fibers. Now, units. Now, when you take a nerve from this muscle and you use it to reinnervate the biceps, the lateral head, you see that the muscle composition changes. So you don't have type 2 fibers anymore, but you have a composition similar to this one. So when you have small units, usually you are able to grade the force much more finely. So we believe that this could be important for prosthesis control because, it's very important that you can modify the force with very big precision. So what we are doing at the moment, again, we are collaborating for electrophysiological recording with the group of Professor Eisman, and the idea now is to actually reinnervate a muscle with multiple nerves and try to understand if we are still able to detect the activity coming from different nerves. And this is the first preliminary report by Matthias Luft, who actually used two nerves of the upper limb to reinnervate the biceps. And we did our electrophysiological recording. And this is a study in progress, but we are actually able to detect the activity coming from the two nerves. So hopefully you will hear about the result of this study soon. Always related to prosthesis control, you know that when a person loses their arm, for instance, not only they lose the capability of perform a function, but also they cannot get sensory feedback. And the sensory feedback can be, the original sensory feedback can be substituted via artificial feedback, for instance, using electrical stimulation, using vibration. So the idea was, why not to design an electrode that can record the EMG signal so that we can understand which muscle a person wants to activate, so which function of the prosthesis they want to activate, but also that contains stimulation electrodes so that we can actually provide sensory feedback. And we had a simple task where we actually ask, so I'll actually go back. So we have an electrode close to the skin. This is a stimulation electrode and one within the muscle. And we stimulate with two different levels, low and high, in stimulation site 2 and stimulation site 3. And then we ask the person to guess, did you receive a stimulation in electrode 2 with low intensity, or in electrode 2 with high intensity, in electrode 3 with low intensity, or in electrode 3 with high intensity? And what you can see, is that usually they were able to guess, to understand where the stimulation comes from. There are some mistakes, of course, but the element in the diagonal are the ones that are darker, meaning that in most of the cases, they were actually able to understand if the stimulation comes from within the muscle or from the skin. So this means that with one electrode, we are able to convey or like to have two channels of stimulation, two channels of sensory feedback information, which may be important. These are, of course, very preliminary results, but I think there is an important point that we can make. Invasive measures are always invasive, right? So if you want to go for an invasive interface, you want to have surgery and have this interface only when you are very sure that it works. So if we have an electrode that we can use in acute experiments, so we can insert, right, if it doesn't work, modify the design. I think this is a very important aspect because, you know, we can improve the design with many iterations until we believe that this technology is ready to be implanted. So even if there are preliminary results, I believe that they are important. And finally, another example that you have seen from Alejandro yesterday as well, so I will be very fast. So we have used the electrode specifically also for stimulation and recording, also in the reduction of pathological tremor. So we target people with tremor, wrist tremor, interflexion and extension directions. And in this, the method was proposed in the first study by Steiner Rosen. So the idea was, why not to stimulate? I will actually do that. So we use our electrodes for both recording and stimulation. We place them in the flexor and extensor muscles of the forearm. And these are extensors, and these are flexors. So the idea is that before we record the signals, and we are actually able to estimate the tremor frequency. And then in the next phase, we stimulate. So when, for instance, the extensor is active, we stimulate the flexor to counteract the activity. And when the flexor is active, we stimulate the extensor. Now, something important and is that doesn't matter if we stimulate above the motor threshold or below the motor threshold. So we are able to reduce tremor in the same way. So like afferent stimulation is actually able to reduce tremor in these people. And I will briefly show just 1 second of the video that you already saw with Alejandro yesterday. So basically, this is before stimulation, during and after stimulation, actually tremor in these people were reduced. And this was actually, I've been very fast, and this was actually all what I want to say about intramuscular techniques for recording EMG. Just summarizing, so I present like different electrodes that have been developed for decoding the activity, motor unit activity in deep and superficial muscles. I show you that we can tailor the design depending on what we want to achieve. And we can use this type of electrodes both to answer fundamental questions like in motor control, but also neuroprosthetic applications, so as a neural interface. And I also show you that it's possible to do acute tests with this invasive electrode without any surgery, so that we can actually optimize the design before making this electrode implantable. I presented many studies, and of course I didn't do this work alone. Like this work started when I was a postdoc in Dario's lab, and he has benefited from the contribution of many partners and many different institutions, and I represented the main ones, but there were also more people involved. And we also like include a study done within a number of European projects. So I just want to provide a summary. And now I'm ready for questions if you have any.